Thank you, Jane. I'm really, really pleased to be here, and I'm thrilled to follow Joe. Really, I don't think many people know this, but I started my PhD in marine science, in geology. That's not the view I want. I think what I'll do, at least it's showing something. Okay. And I started actually in paleoceanography. So I was trained as a paleontologist, and then I moved into molecular genetics. But I still like to look at this historical perspective, and it's wonderful to follow the sort of 10,000 year view of the bay, and now we're going to the sort of hundreds of years view of the bay. So I've always been intrigued by looking at the historical perspective. When I came to the biolab in 2004, I came across the fact that this was a bay that a lot of people had done early natural history work in, including Proctor, who I'll get to. I just want to add one thing. It's funny that we used to, 20 years ago, I was collecting with a colleague who was then at Harvard in these deep holes in Belfast, off of Belfast, and we were looking for this very unusual, rare annelid worm, and now I know why those holes exist, so that's great. And they've really unique fauna down in those deep holes. We did it through the University of Maine using the Darling Center's vessel, and nearly froze to death. We did it in January, about 20 years ago. Okay, so what I'm going to talk about today is a multi-year project that I've done with a whole bunch of collaborators, and looking at what's been in the bay and looking at fisheries. I'm not a fisheries person. This is not something that I get to spend a lot of my time on, but I'm really glad to be here. By having Jane invite me, I've been forced to sort of pull together lots of things. So one of the key things that came about in 2009, this paper, which you may not be able to read the title of, came out. Let me see how this works, Chris. Okay. In Fish and Fisheries, this was the history project run at the University of New Hampshire with Karen Alexander and Bill Leavenworth and others. And this figure is from their paper, and what this shows is in 1861, the logbook and where folks were catching cod. And if you add these up, this single vessel fishing out of Trenton up in here somewhere landed over 10,300 cod in a single year. It shows where they were fishing. And you can see right off of Bald Rock, and I'll show some video in a minute of what Bald Rock's bottom looks like today. It was productive, without a doubt. The reason this data exists is because from about 1790 through the Civil War, the federal government actually gave money, economic development money, cod bounties out to fishermen. They really supported the fishery, and it's sort of like what we'd see today, the pine tree zones where people want to start businesses in Maine. The cod bounty was similar. It was an economic development exercise. So the fishermen kept incredibly good records, and one of the best places where these records exist is in Frenchman Bay. The archives are thick with single vessel records of how they fished, who was fishing, and what they were catching. And so this paper is an intriguing read about the manpower hours required to do this kind of work and who was actually doing the work. Great read. Fast forward, this is 1861, this is August of 1935. These are pictures that my grandfather took. This is Tud Bunker from Cranberry Island, and you can see that well into the last century, there was ample cod in this bay. These, I think, were caught on the Baker's Island ledges, and this was sort of a day excursion. And you can see they're using the classic tub trawling methodology that developed in the late 19th century and came into the 20th century. More on that later. And you can see the yield was fairly good in one afternoon of fishing. I do want to point out that down here, these are from a scrapbook I have at home. And I pulled these out last Sunday, and I was looking at them, and I was thrilled to see this. This is a barn door skate, which I think is locally extinct now and is essentially gone, which is too bad. And there's another one, if you look carefully, laying in the bottom of the dory here. So we have this historical record, and when I came to the biolab, as I said, in 2004, I came across the Proctor Biological Survey of the Mount Desert region. And Proctor is an interesting character. His grandfather had founded Procter & Gamble. He worked at the biolab in the early decades of the 20th century. This is him standing outside of what I think is now what we call the Epstein or the Instrument Shed Lab.
Forster Cottage.
Is that Forster Cottage?
Yes.
Yep. Thank you. I wasn't sure. But he worked here until 26 or 27, and there was some kind of falling out that I haven't really dug into, and then he moved his whole operation to his home property, which was on Eden Street. And for many years, he collected in the Bay. I think he collected at 153 different dredge sites across the Bay, as well as shore sites, which were hand collected in the intertidal. And most of that collection, after moving around New England a little bit, spent some time in Boston, then at UMass and Amherst, is now up at, in the Saltel collection at the headquarters, which is great. He actually, at age 35, he'd been an investment banker, and at age 35, he retired. He didn't really need to work, and he went back to school at Columbia and was trained in the department that included a lot of the preeminent biologists of the era and geneticists. So his training was quite good at Columbia and was given an honorary degree. He was not the first guy to work in the Bay, though. Dwight Blaney, out of Ironbound, did a lot of work, particularly on mollusks, and in 1904 published a paper which is unique, I think, and pertinent here, in that he looked in cod stomachs and discovered, just looking in, not cod, excuse me, in haddock stomachs, in 10 haddock he found 26 different species of mollusks that haddock had been eating, just to give you some idea of what was on the bottom then, and what these groundfish, which we don't see commonly anymore, what they were eating. And then obviously the biolab for the greater part of the 20th century was also collecting. And the collection records are in my office. I've started going through them. And what I'm trying to do, sort of the goal is to say, we have the fishing records from the 19th century, the cod fishing records. We have some early work by Blaney. We have Proctor's survey. We have work through the 30s to 60s, lots of it by Hans Borai. And then what's the bay look like today? We do know that the ground fishery has collapsed. So that's sort of the background of where I came from. Obviously, you can add on to this Ted Ames's work. So this is actually taken from Ted Ames's narrative history paper on cod, where he published, and this is Rich's map of the grounds, and you can see here's Mount Desert Island. And they don't really describe what's going on up in the bay, but you can see Martin's Ground, which I think is actually a little closer to shore, and these Baker Island ridges, which were fished heavily. But obviously in that previous slide, you can see they were hauling a lot of cod from up in here by Bald Rock, in the Upper Bay as well, over by Calf Island as well. So in 2010, I've been teaching actually molecular genetics, and I'm interested in how fish populations move around, and I use molecular tools to do this, but I've always sort of been interested in this historical perspective. And in 2010, I met Brian Tarbox, who is a commercial fisherman, former commercial diver, worked for the state, I think, diving under aquaculture facilities, and now teaching at Southern Maine Community College. And he and I have been teaching population genetics to the SMCC students. And we started teaching, I think we were working that first year doing population genetics of smelts, and we have mutual interests. And Roger said, you know, you need to, Brian said, you've got to meet Roger. Oh God, I'm having a brain cramp. Roger Woodman, thank you. Roger Woodman, because he's really interested in the fishery. Roger ran a commercial fishing venture out of Portland for many years with several vessels, fishing Gulf of Maine and over into Canada. And he had bought this schooner, Alert, which actually was derelict at the Jock Williams Yard in Somme Sound, formerly owned by Thurman Radford, and refitted it to go fishing. And you can see here the tub trawls. And so we went out fishing and decided, knowing that we had this 2009 paper on these cod grounds. We wanted to see what was on these cod grounds. But rather than go out and drag them with a dragger, we should go out and fish them the same way that they would have fished them 150 years ago. And I'm going to try and play some video here, if I can do this. Okay, here we go. Uh-oh. So what? I guess I'm not going to play that video. Let me see here. Maybe I need this to work. I had to buy this on the iTunes. No, I'm not going to play this video. What I'll do is I will I'll get this video running at the break out in the in the linker out here. Okay. So fast forward, we did the fishing. We put thousands of hooks on the bottom. We used a whole bunch of different kinds of baits. And I think over the course of 5 days, we caught maybe 15 fish. 4 dogfish, 2 skates, 1 39-inch halibut, no cod, some hake. It was discouraging. And the video I'll play out in the linker at the break shows what the bottom looks like. We also used a Scottish seine, which is a simple net you put out, circular, you haul back fairly rapidly, and we've got good footage of that coming in. We caught one winter flounder about this big. So there aren't a lot of fish out there. The bottom looks pretty good. The video is fairly good. The nice thing is we have high production quality, because what happened was sometime in the winter of 2010, PBS called me here at the lab and heard about what we were doing, probably because of this article in the Bangor Daily News. I don't know exactly how PBS got wind of this. And they said, how do you feel about us coming and putting a film crew on board for 2011? I called Roger and Brian, and we got a little bit of money from the Maine for the small campuses across the University of Maine system, a $10,000 grant. And we went back out in 2011 with Carl Safina, and that show called Cod Comeback, as part of a series called Saving the Ocean, has about a third of the episode is about Frenchman's Bay. And it's great footage, and it helps to have a professional film crew on board. So some of the other things we did in those years is this is a transect showing temperature across the bay, and the horizontal scale is shrunk. This is the mouth of Jordan River with a temperature about 64 degrees. We were running equipment through the bay. This is Egg Rock out here, and the temperature out here is in the 50s, about 52. We also ran a multi-beam sonar, and you can see here, and the light's not very good, but what this does is images. This is a little school of fish. It's about 6 meters off the bottom. It's about 20 meters long and about 4 meters wide. So there are fish out there, but they weren't, we weren't able to pick them up using the traditional techniques, which was too bad. Okay, so I'm going to go on to 2013. We were supported from the Davis Conservation Foundation to go back out and look at these grounds again and compare the records of what Proctor had collected from 26 to 32 to what we could find today. I got Brian Tarbox back. He brought this small, remotely operated vehicle that has a video camera on it that we ran onto the bottom in July of 2013. The way this works, I had 2 students helping me. You'll see them in a second. One was Shannon White, the AmeriCorps volunteer. The other was Liz Thompson, who's an undergraduate at SMCC. She was funded by the NSF, and essentially the students were down below driving, sort of like a video game, driving the ROV and trying to get video camera. And we record the video on a computer just right on board. The other thing we did is we sampled all these sites, and this was a preliminary investigation. We sampled 7 sites. We just take a small dredge. We were doing it by hand, sampling across all these areas, and then we bring the samples back, and this is a sample. This is probably very much like what Joe was talking about off of Sand Beach. This is off of the south end of Turtle Island near Winter Harbor, and it's very, very coarse grained. material, but we also sampled in the mud of Frenchman's Bay and pulled out all the organisms that we could find sieving at different sieve sizes. Here's Elizabeth and Shannon. We couldn't have done this work without them. They were incredible and persevered through lots of mud samples. Okay. So one of my goals is to actually see what's down in the bottom, and this was made a lot easier. This is the Proctor data, and this was put together, I think, by Alex Brett at COA, and this is part of the Frenchman Bay Atlas. I think this is part of the outcome of the Frenchman Bay Partners. It's wonderful. What you see here, obviously, is the bay. This is the Bald Rock site from Proctor, and this is Alex has taken the species names and put them in, so you can actually click on the site, and you can see what Proctor found when he collected there. We've done the same, and I'll show you the sort of comparative data in a minute. And then this is what the bottom looks like. And I'm going to play a little video here. This video isn't going to work because I was able to embed it on my computer. Unfortunately, when you have PBS, you can't really edit that video. They own the video. I had to buy it. So that's why the other video didn't work. So we'll just let this play. And these are edited. So this is the bottom of Bald Rock, just to the east in the same spot that Proctor collected. The red dots you're seeing, we have 2 laser pointers. You can't always see them. And they're to sort of scale how big a frame of view you have. We're seeing some hermit crabs here. The bottom is not very diverse. This is right off the edge where you get off into the beer bottle, encrusted. We tended to go to each of these sites and look on the ledgy part and then down where the bottom flattened out. So I have some other video from Bald Rock. This is just off the deeper side. What you're seeing here is actually the heading on the ROV, so which direction it's looking. When I edit the film, you lose the depth, but the depth here I think is about 9 meters. Same thing here. So this is Calf Island, another site that Proctor sampled. I will say that we preliminarily sampled 7 sites in the bay. The reason we did this and how we selected the sites is any site that Proctor had sampled in the 20s and that had been a historic fishing ground in the 1860s was automatically on the list. I think 1 or 2 of these sites we also went to were sites that were very high yield ground fishing sites, but Proctor might not have sampled there, and that's the case with the Martin's ground, which you'll see in a minute. So this is Calf Island, the channel right here off the south end of Calf Island. Again, Proctor's list of species. And this is similar. Wait a minute. Sorry. I was talking to Ed this morning, and I think it would be nice to go into some of these shallower sites at least and do a lot of this video by scuba. The ROV is effective, but depending on the current, this site had a lot of current as the tide was going out, and we found that it was hard to film. And you can see again, not a lot of diversity. There are scallops at this site, not in high density, which you'll see in a second. Burrowing anemones that are going to get scared of the ROV. And then as you go up into the shallower part, so this is going from the deeper into the shallower part, you're now onto the hard ledge, and you can get a sense. Shannon has looked at all this video in excruciating detail. You can see the scallops there, down in here, lying on the bottom. And we've identified, to some extent, as many organisms and species we can from the video footage. The grab data and the dredge data is a little bit more, you actually have the physical animals or pieces of them, and you can see what you get from the bottom. So that's Calf Island. Turtle Island was a really interesting site. Turtle Island, for those of you who don't know, is down here, right off, this is Winter Harbor. This is Turtle Island right here, and this was a fishing site and as well as a Proctor site. And we went from the shallow water, let's say 50 feet, and then dropped down into 120 feet. And you'll see a significant difference from the rocky shallow site here. A little more diversity. This looks like an encrusting tunicate that's actually invasive, we think. Horse mussels, modiolus, are pretty common, both dead as well as filter feeding normally. Interestingly, we don't see fish, essentially. That may be partially artifact of the fish being spooked by the ROV, but the ROV is not very big. You know, it's box-like, it's about this big. I don't think it makes a lot of noise, but I think over the course of hours and hours of video, we saw one cunner. That's it. Little, like this.
What about mammals? Any curious seals?
No, nothing. I mean, I think we saw some mammals during the day. Then when you drop into the deep water, we were sort of surprised to see this. Common sand dollars, just fields and fields of them. We dredged into this site, and in a single, 30-second dredge, came up with 180 sand dollars, and almost nothing else. This is very coarse sediment. Obviously, it's south-facing. Lots of wave action coming in. It's not protected at all, and we were sort of surprised to see this. Interestingly, and you'll see, in just a flat text file at the end here, Proctor didn't even record common sand dollars at this site. Now, that may also be a collection artifact as well, which I can comment on in a minute. And you can see here are, well, you could see the two laser pointers sort of as a scale for us. And then this is Martin's Ground. Martin's Ground was not collected by Proctor, but is a fishery site and was recorded on that old map by Rich. I have a chart in the back if you want to know. This is just east of the Schooner Head, Sand Point area. And I think Ed knows this site well. He has his own name for it, which I won't divulge. But this is a cool site. This is, I think, about a couple acres. It's about 50 feet deep. It's surrounded. It's surrounded by deep water. 100 feet deep, or over 100 feet deep, and then there's this knob, sort of like a monadnock, if you will, where it's just a knob coming out from the floor. And this is quite diverse, and especially in the cnidarians. So these anemones, this was sort of a hard site to find with the ROV, but it wasn't too bad. I think it's probably a great site to dive on as well. And we've recorded all these species using a couple different techniques. Okay, so what do we take away from this, from both the video as well as the dredge data, is a sense of trying to compare data between what Proctor was finding and what we found in 2013. And you can look at that in this sense. So here's the list, for example, at Ball of Rock of the species, and these are updated to modern taxonomy of what Proctor collected. And here's, wow, I put 2014 there. We collected in 2013, that's my typo, I'm sorry. And you can see here, for example, what we found. And we don't see a lot of overlap here in green. This is a small bivalve Wacoma that we picked up, that he picked up. I don't think he dredged cod in knowing the methodology he was using. He's obviously not here today to talk about exactly what they were doing, and the descriptions of how they dredged are okay, but not wonderful. I think they were probably fishing as well as dredging, but he doesn't come out and say that. And you can see here, back to one of the first slides, the taxonomy of this species is changing, but I think it's Dipterus laevis, the big barn door skate. It's now extirpated in Frenchman Bay as well as most of the Gulf of Maine. He was finding it at several sites. They were finding cod, they were finding winter skates. We're finding essentially none of these. And you can see what we've found. One of the other things I did, and this I did with, again, Brian Tarbox, and we did this as a student exercise with Southern Maine Community College. We took samples that we were unable to identify, or partial samples, and ground them up, made DNA in early January, and then sequenced a certain gene to try and figure out what they were. This was reasonably effective, and we picked up a lot of different species. One of them, this invasive tunicate, Ascidian, that's white and encrusting, and we're picking it up we're seeing it in the video, we get it in the dredge, but we have techniques, including ROVs, including DNA sequence analysis, that we can use to identify things that Proctor never would have had, obviously. But you can see there's not a lot of overlap. I will say also that I've just started going through the MDIBL records, and what my goal is to fill in at these sites. So at Bald Rock, that's a site that was collected heavily by the biolab over the years. And I have all those collection records, but to tell you the truth, I haven't gotten to them yet. Just one more. This is, what happened? That's odd. This is not Bald Rock. This, oh, I know what it is. Ignore this band here. This is actually the Turtle Island site, and you can see we are seeing some overlap. This mollusk tends to appear quite a lot. We dredged it a lot. This ocean quahog, it's not probably commercially viable. It's a fairly small bivalve. But you can see we're not seeing a lot of the same things. I actually think this may be collection artifacts more than anything else, but I can't, it's hard to prove that. And finally, this is from the Gulf of Maine Research Institute's white paper they put out on cod in June of 2013, and you can see, and these are populations that Brian Tarbox and I and others have done population genetics on, and these are fairly, these are genetically recognizable groups of cod, and there are cod being fished off of Jeffrey's Ledge in a small way, south of Portland, but as you can see here, this is, our data confirms what they know from cod tagging studies and catch studies, that these inshore grounds that were really well described by Ted Ames, these fish are gone, and which is really sad, because what was a very productive inshore fishery is gone. And the question that will come out of this work eventually is, has the benthic ecology of Frenchman's Bay changed drastically when you take away this guild of fish, including haddock and cod, and to some extent halibut? And I can't imagine that if you had these organisms that were apex predators feeding in these bays, that you take them away and the bay is going to be different. We can't prove that yet, but that's sort of the ultimate goal of these studies. I'll refer back to Dwight Delaney's 1904 paper. If they were seeing 26 species of bivalve, or not bivalve, of mollusk in haddock stomachs, you got to know that taking away haddock and cod is going to have an effect to this bay. We can't characterize it yet. And I think I'll stop there for questions.